Hello and welcome to the first episode of our antibiotic guide lectures. We will start by looking at the pathogenesis and the microbial element of antibiotic therapy. We should analyze how bacteria work in general, just a small introduction in our first lecture. So, we will talk about the pathogenesis of the microbial invasion. It all starts with the adhesion. The adhesion, which is the first step for the bacteria to get inside our body and our cells to be able to infect them. So we start everything by a bacteria that is flowing around. It enters either through the skin, through the body, through eating, through our nose, but for it to have any effect, it should stick, it should invade. So first of all, it should attach. And for attaching, we should have specific antigens on these bacteria. These specific antigens should have specific receptors on the host cells. That's why it is a mandatory prerequisite. If these bacteria will have, for example, specific antigens that have receptors only in cows, for example, this will be a bacteria that can make infections for the cow. It should have human receptors. And usually these receptors and ligands are either on open skin or inside endothelial cells or epithelial cells. Most commonly, it is in epithelial cells. If we take, for example, any bacteria that invades our upper respiratory system, for example. The second part, it's the invasion part. And as we can see, the invasion usually happens with endocytosis, to penetrate the cell and get deeper inside the tissues. The next step usually is destruction of intercellular junctions, so that when the bacteria, the first wave of bacteria enter inside the cell, they destroy the junctions between the cells. So the first barrier, which is the epithelium, for example, becomes obsolete, and they bypass this primary physical barrier. The molecular invasion strategies, we can have two examples here. We can have the Yersinia, for example. The Yersinia directly binds into integrins, which are the human receptors, to the antigens of the Yersinia itself. This, inside the cell, will give us a reaction where the actin filaments will start to engulf this cell and get it inside. This how the Yersinia invades. This is a pure receptor-mediated entry. Or if we will have TTSS effector translocation, it means that when the cell binds to the receptor, as in Salmonella, for example, we will have different types of proteins that are getting inside the cell or activated by these antigens. For example, the SIPE will activate the actin to start this splash, which is engulfment. This is similar to the receptor mediated, but we add to it the SOPB and the SEIPA. These two proteins will have the effect where the actin filaments will start to close up irreversibly. This allows them to have faster entry into the cell. Another strategy that is similar, another example of translocation of TSS effectors is the Shigella. The Shigella will have depolymerization of microtubules and of actin using its own type of antigens and receptors. They will engulf fast, and the Shigella will be able to enter or to enter its own receptors. We can have here Listeria, which have both pathways. It can have a pure INA, which is a cadarian, which is a typical type of receptor. Or we can have the second type, which is also by polymerization and actin depolymerization. As we can see, we have the first receptor mediated and non-receptor mediated types. Here we have Listeria having the dual pathway. If we can have a study, a case study, it's uropathogenic E. coli. First of all, we have the lumen. It's in the lumen of the urinary system. We will have replication, then adhesion. The adhesion will be on mannose receptors that will attach to the pili antigens. Using this, the bacteria gets inside the cell. The urine continues to flow, and we will have intracellular replication, not only luminal. This intracellular replication will attack the structure of the cell, and we will have exfoliation of the cell itself. This intracellular bacterial community is a big part of the persistence of E. coli. This is how they persist inside the cell. Then this exfoliation and this gap that happens here will allow the E. coli to get further inside our tissues and maybe in the blood. We have a different type of exfoliation that is chitosan induced, which means that when the bacteria is inside the cell and it feels the attack of the immune system or antibiotics, it will have F-actins around it to engulf it. Then when we will have this exfoliation, the intracellular bacterial community can burst and we will have resurgence of the infection once again. These can happen, for example, in two steps. The first step is the first infection, we take antibiotics, we have dormant E. coli, then we have the second phase with the resurgence. If we will talk about bacteria in general again and their pathogenic characteristics, we have obligatory extracellular. These bacteria multiply only outside the host cells. For example, pneumococcus. Or we have intracellular facultative. It's when the bacteria can have outside of the cell replication and inside of the cell, as we saw just with E. coli. This will be slow evolution. The bacteria will replicate slowly inside the cell, but will have frequent reactivations and they can evade the immune system inside the cells. And for these bacteria, we need antibiotics that can penetrate the cell. Then we talk about intracellular obligatory. These are cells that live only inside the bacteria. The bacteria that only lives inside the cells. They only multiply inside. For example, we have chlamydia and all rickettsias. How do bacteria attack us? Usually using weapons. These weapons are toxins. They can be exotoxins, which are molecules that are secreted. They are secreted proteins that get secreted from the bacteria inside the bloodstream, inside the local tissues, and usually it is not destroyed by antibiotics. These toxins cannot be destroyed by antibiotics. So if we have bacteria and we kill it by antibiotic, the toxins will stay until their effect goes away. Examples are tetanus, diphtheria, and botulism. Or you can have endotoxins. For example, all gram-negative bacteria have a thick wall. This thick wall have lipopolysaccharides, which they themselves can activate the immune system, and they can induce bacterial lysis. No, they are induced by bacterial lysis. Like when the bacteria gets destroyed, we have bacterial lysis, and they will get into the bloodstream, all the local tissues, and will give us a inflammatory immune reaction. Then we get to the superantigens. We will get into immunology later, but usually to get an immune response, the bacterial antigen should get inside one of the phagocytes or the macrophages, and these will alert the immune system to start activating that cytokines. But sometimes we have superantigens. These superantigens, they bypass the TCR, they bypass all of them, and they bind directly to the TCR. They bypass the recognition by other cells. So this triggers immediate massive immune activation. For example, we have the staphylococcal effect. That's why we have really big intoxication in case of staphylococcus. And this will induce a cytokine storm. We will get into cytokines in our next lecture. But these proteins are like the signals, and they will result in systemic inflammation. The clinical presentation of big cytokine storm is sepsis, which will give us endothelial dysfunction, increased capillary permeability, and multi-organ failure. We will get into this inside our immunology section. Then we will get into biofilm. The biofilm is when we have a material surface. It can be our material, or it can be the most common place for a biofilm is non-human material. For example, a catheter, a changed valve. So the bacteria gets stuck into the surface and interacts. Then we will have proliferation of a colony. These bacteria will produce a film. This film is made by peptidoglycans, and it envelops the bacteria. This will protect it from human cells and from antibiotics. This 3D community will give us a physical barrier that doesn't allow antibiotics or immunity to get there. We have specific antibiotics that can pass through biofilms, but this should be taken into consideration when we are dealing with biofilm secreting bacteria. Another persistent mechanism is small colony variants. For example, we have Staphylococcus. We have thousands of them. Then we get any kind of stress, which is hypoxia, for example, antimicrobials, immune reaction. Then we will have small colonies. They have slowed metabolism. They are really similar to spores, but they are still alive. They are not spores. They are cells that have reduced metabolism and increased resistance, and they are really slow. And then we will have two types. The unstable, these ones that will have switching phenotypes. Usually these are starting to get resistant. And then we have stable SVCs. These stable SVCs can stay for really long time. This formation of a biofilm and the persistence in cell will give us changes inside the cells themselves in these SVCs, SCVs. And then when we get a favorable condition, they will pop again and they will give us another infection. If we go beyond bacteria, then we will have viruses, for example, which have a direct cytopathic effect on the cells. We will get into viruses more inside the immunology section. Fungi, they can have invasion of the cells, but with different, it's more of a mechanical invasion. And then parasites who get fixed in between. So if we get into a summary, it is adhesion and entry, change of cytoskeletal, so we can have the endocytosis. Then we have the chemical modulation, where we will have endotoxins, exotoxins, and the immune reaction. And then when we have the immune reaction, we will have the strategic persistence, which can be inside the cell by biofilm or small intracellular, small colonies. The small colony variants. This is the end of the episode 1, and thank you for listening. We will see you again in our next episode next week within the bacterial guide of antibiotics. Thank you.